Welcome back. In this section, we'll be talking about two-hit hypothesis and genomic instability, and really where those mutations come from in the first place. So you can acquire mutations two different ways, either through somatic mutation or germline mutation. Somatic mutations are acquired by somatic cells, which are all the cells of the body except eggs or sperm. And those mutations are passed on to daughter cells during cell proliferation. It is important to remember that these mutations cannot be inherited by offspring. Every time a cell divides, it must replicate its DNA, and errors just get made by chance. So you'll remember what we talked about before with a lot of DNA packed into a very small area. So just by chance, about one mutation is made every 10 billion base pairs. There is an increase in DNA damage and subsequent replication error due to environmental carcinogens as well. These include UV damage, which is also shown in the cartoon to the right, where an incoming UV photon hits the DNA and damages it so that it causes nicks in the DNA, which you can see to the far right, which then have to be repaired by the cell. And this can cause an increased risk to skin cancer. Similarly, smoking causes chemical damage to lung cells, and hepatitis and alcohol abuse can lead to damaging cirrhosis of liver cells. You might remember these from another lecture where Dr. Pienta discussed the risks to different cancers. The other way you can get mutations is by inheriting them, and these are called germline mutations or inherited mutations. And these mutations are present in the germ cell, so in this case that's the egg or the sperm, and are inherited by offspring. So it's just the genetic variation that we're born with. Germline variation accounts for why offspring look similar but not identical to their parents. When cancer is said to run in families, it may be due to these inherited mutations. For example, women who inherit a BRCA1/2 mutation are at increased risk for breast cancer, and individuals who inherit a CDH1 mutation are at increased risk for stomach cancer. But not all women who inherit a BRCA1/2 mutation develop breast cancer. How does that work? This is where it becomes important to remember that cancer is caused by an accumulation of detrimental variation in the genome. The biggest risk factor for developing cancer is aging. Over time, we accumulate mutations, and this either happens by random replication error or by increased replication error due to carcinogens that cause DNA damage, as we discussed before. The two-hit hypothesis describes why not all women with a BRCA1/2 mutation get breast cancer. You'll remember that humans are diploid. We have two copies of every gene, one maternal and one paternal. This means that even if one copy of a gene or allele is mutated, the other copy can allow the protein to operate normally. In order for a gene to be cancer-inducing, both copies of the gene must be affected. The second so-called hit may alter the DNA directly via mutation, or it may alter the expression of the DNA via an epigenetic mechanism. So let's walk through an example of this. We'll walk through the top tree first. On the far left, you can see a parent who with his partner passed on two normal copies of a gene to their child. Over the lifetime of this individual, he accumulates mutations, including one hit in the gene shown here. Importantly, the other copy of the gene that was normal, but did not have a mutation, allowed it to be expressed normally. Eventually, however, this individual acquired another mutation in the same gene, and this resulted in the gene becoming a cancer-causing genetic mutant. In the bottom example, we see an example of inherited or germline mutation causing increased risk to an individual over a lifetime. In this case, the parents passed on one mutated copy of the gene and one normal copy of the gene. In this case, as the individual progresses through life, he only needs one additional hit in order for the gene to be cancer-causing. Most cancer requires mutations in multiple different types of genes. In order to survive, a cancer cell must overcome normal regulation of cell proliferation, cell survival, and cellular communication, and other so-called hallmarks of cancer that will be discussed by Dr. Zarif in a later lecture. Each of these processes are tightly regulated in normal cells by multiple redundant pathways. This means that in order to become cancerous, a cell must accumulate mutations in multiple of these pathways. It's been described as usually a minimum of 6 to 7 in multiple different genes. So you can see an example of this to the far right. At the top, we see a normal cell where a mutation has inactivated a tumor suppressor gene, so we see increased proliferation. This cell doesn't become malignant yet, though. First, it undergoes mutation that inactivates DNA repair genes, an additional mutation that activates an oncogene, and then mutations that inactivate several different tumor suppressor genes that then induce it to become cancer. In addition, cancer cells show a great deal of genomic instability. And genomic instability describes how cells can survive and divide with higher rates of mutation than in normal cells. So in a normal cell, DNA replication error results in programmed cell death or apoptosis, which you can see in the lighter colored box over to your right. In a cancer cell, that same DNA replication error with the same DNA replication error, cells continue to divide and pass that mutation on to daughter cells. Over time, these mutations in these repair pathways will accumulate since cells never undergo that programmed cell death, and the frequency of mutation in the cancer cell genome increases. This increase in mutation rate leads to tumor cell heterogeneity and is one of the reasons why cancer patients become resistant to treatment. This brings us to the end of our lecture. I hope this gave you a good understanding of the basics of genetics in order to better understand the rest of this lecture series, An Introduction to Cancer Biology.